Now, meiosis is going to be pretty similar to mitosis, but a little bit busier and a little bit more physiology to understand. So, meiosis is basically sex cells, or the production of sex cells, I should say. Somatic cells, which we just went over with mitosis, have 46 chromosomes, 23 pairs of sister chromatid. Another form of cellular division is meiosis, which only occurs in the gonads. These are your sex organs, your testes and your ovaries. And this is going to be reducing the number of chromosomes by 1/2. So instead of 46 chromosomes, we'll have 23. No pairs. Meiosis is producing our sex cells, so what we call these gametes. All right. Male gametes are sperm cells in the testes, and female gametes are egg cells in the ovaries. And again, these gametes have 23 chromosomes. So gamete formation in humans is categorized as the egg producing 23 pairs, having the egg producing cells in the ovary having 46 chromosomes and the sperm producing cells having 46 chromosomes, but then they undergo meiosis, and then now they have 23 unpaired chromosomes. They have one set of chromosomes, all right? So they don't have sister chromatid. They come together, one egg, one sperm, to fertilize and create a zygote. That can become its own individual because it has 46 chromosomes, 23 pairs, 23 homologous pairs. We can kind of see it represented here, diploid being zygote, or its own individual organism, 2N, N being haploid, right? N being haploid. We can have, this would be a functioning being. Well, after meiosis, we only have half of that genetic material. Okay, so we have 4 here, we have 2 here, 2 here. Those can come together and form another individual with 4 total again. All right, so individuals that are functioning, not individuals. These can come together to become an individual, though. So we have 23 pairs of chromosomes. There are 22 pairs of autosomes. These are non-sex chromosomes, so you can kind of see those represented in this karyotype. So these are all the different sister chromatid, okay? 22 pairs of those. And we have 1 pair of sex chromosomes. If you're a male, you have an X chromosome and a Y chromosome. If you're a female, you just have 2 copies of the X chromosome. And this is the basis for genetic variation, is these different chromosomes coming together and exchanging genetic material throughout meiosis to form variations in genetics. And so when we're talking about, when I said homologous, you're probably, oh, what did you just say? This slide kind of explains that a little bit. And so homologous pairs of chromosomes basically mean that those 2 chromosomes have the same size and shape, and they also carry the same genes in the same locations. So we have, this would be 1 pair, so this could be like chromosome 1 or chromosomes 2, right, homologous pairs. The genes represented by these letters are the same locations, okay, same size of the chromosome, same locations, but we can have different flavors or varieties, if you will. And these different versions or flavors of the genes are called alleles. And so this could be kind of like blue eyes or brown eyes. We'll get more into this after exam 1 when we start talking about genetics more. This would be an example of a non-homologous pair of chromosomes because these are not the same gene. The same chromosome, and obviously not the same genes. So meiosis is going to be happening in the sex cells, in the ovary, and in the testes. It results in 4 daughter cells with half the amount of genetic information. So we start with 1 cell, has everything, splits via mitosis, and then it continues to split into 4 haploid cells with half the genetic information. So genetic variation among offspring is created via meiosis. Meiosis and fertilization are responsible for most of the variation from one generation to the next. And there are millions of possible combinations of genes that each parent can produce due to 3 different concepts that we're going to talk about: crossing over, random alignment of those homologous pairs, and random fertilization. So with crossing over, we're talking about homologous pairs kind of trading parts. And so homologous pairs in prophase 1, as you can kind of see here, they kind of sit next to each other. So just like we're talking about in prophase of mitosis, those homologous chromosomes condensing, well, that happens in prophase 1 as well, but they're right next to each other. Homologous pairs are next to each other. And so they can kind of exchange information called crossing over. And this can occur several times in each pair, such that we can have these crazy arrangements. We can be exchanging different parts of different tails. So you can kind of see this is the way that the homologous chromosomes line up next to each other. This is how much they swapped. Like this green came from over here, this red came from over here, and then the oranges and yellows swapped, and all these genes are all over the place. And this leads to a ton of different variations, such as brown hair, blue eyes, and big feet, and smelly breath. But you know what I'm saying, like tons of different stuff in the population pool that can be sampled from just this one event. Random alignment does basically the same thing, just at a different stage. And so instead of changing genetic information, we're changing the alignment variations that we can have when those chromosomes line up in the middle. So this happens during metaphase 1. So we're literally randomly aligning these chromosomes in the middle of the cell and then splitting them accordingly. So we're in metaphase 1 of meiosis 1. Okay, so what we're seeing here is metaphase 1. We can have it in this assortment, blue on both sides, blue on one side, red on one side, or we can have it in this way, blue and a red on this side, and then a red and a blue on this side. And we split those, we can lead to different combinations. These are 4 different combinations based off of this different alignment, this one different alignment up here that we had. And this is called random alignment. And so this leads to genetic variation. So how many combinations are possible? Well, for random alignment in particular, the possible number of gametes is 2 to the n, where n is the haploid number of the organism. Well, for humans, it's 23. And so there are 2 to 23, or about 8 million possible combinations of chromosomes. So random fertilization by an ovum, or of an ovum by a sperm, is what can be leading to more variation. All right, so this is that third concept. So any sperm can fuse with any egg. Well, an ovum is one of 8 million possible chromosome combinations based off of our random alignment and our crossing over. Same thing with the sperm, 1 of 8 million. Well, the resulting zygote is literally 1 of 8 million times 1 of 8 million, or 2 to the 23 times 2 to the 23 possible combinations. And so that's 1 in 70 trillion possible combinations that we can have. So that's a lot of different traits that you can swing around. And crossing over is going to add even more variation. So those 3 concepts are going to be the basis for genetic variation and why you look different than your sister, or why you look different than your mom. Lots of different flavors of genetics. So the summary of mitosis and meiosis is basically that both are different types of cell division, and they occur in different types of cells, and they produce very different products. So when we're talking about mitosis, it occurs in somatic cells, and occurs in skin cells or muscle cells or nervous cells, and produces identical copies. Whereas meiosis is in the gonads. So if you're a male, in the testes, if you're female, in the ovaries, and it produces 4 separate sister or daughter cells, I should say, and those are all half as many chromosomes as the parent cell, and they're not identical. They're all genetically distinct from each other. So that is the basis for our mitosis and meiosis, as well as our DNA synthesis lecture. This would have been strung out over 2 lecture periods with an activity that we were going to do together, but unfortunately, COVID threw things up in the air. So if you have questions with this stuff, please email me. I would love to do this in person, typically, because this is some tricky concepts, I guess you could say. So take it slow when you're reading the material. There's a lot more in your textbook than what you need to know, but it makes sense of things that I talked about. But what I need you to know is based off of this presentation, and questions that I will have in the exam will be based off of the presentation, not necessarily your textbook. So always pay attention to the quiz questions as well as these knowledge checks. All right, so as I go through this, feel free to pause before I reveal the answer so that you can figure it out yourself. So DNA polymerase joins sugars and phosphates together via what type of bond? Dun, dun, dun. Covalent bonds. And can a covalent bond dissolve in water? Heck no. If it could, then you wouldn't be alive when you go in the swimming pool. Your DNA would dissolve in the water and you would be no more. What is the purpose of the cell cycle control? Well, that helps regulate cellular division. What happens when the cell cycle becomes unregulated? Cancer. And when does crossing over occur? Prophase 1 of meiosis. And what about random alignment? Metaphase 1 of meiosis. So don't memorize everything, all the steps of meiosis. All right, understand that it's basically mitosis times 2, and that's why we have metaphase 1 and metaphase 2 rather than just metaphase. Understand the major concepts of mitosis. Don't memorize everything. Don't be able to draw it necessarily. It can help to draw that stuff out, but again, what I needed you to know is the stuff that I presented on these slides. Don't get too bogged down with everything. And again, please email me if you have questions. I'll see you guys soon.